many DNA binding proteins as well. And in fact, those proteins are also part of the genetic material which is very tightly coiled and packed inside a viral capsid. So there also some proteins play a role, right? But those proteins will contain sulfur, but DNA will still not contain sulfur. So sulfur was radio labeled to mark what? To label protein, right? Phosphorus was radio labeled to label
DNA.
yes, nucleic acid, let's say, or DNA, let's say. Now the question that Abhiya's worksheet contains, it says that sulfur is exclusive to just proteins and phosphorus is exclusive to just DNA. Right? That's why that was the basis of this experiment working. You need something to distinguish between DNA and protein. If you use something which is common to both, for example, carbon, if I label carbon, will this experiment yield the same result?
No.
No, because there'll be nothing to distinguish between protein and nucleic acid. Okay, so the same question I have seen coming in a way where if they would have labeled oxygen, if they would have radio labeled carbon, if they would have radio labeled anything else, nitrogen, both nitrogen, carbon, and oxygen, all three of them are present in DNA and protein the same. So you cannot make this experiment work because you remember that when they allowed the bacteriophage to infect E. coli, right? So E. coli was in a way present in the experiment, but that was not the model system that they labeled, right, Fatima?
Yeah.
Yeah. So bacteriophage infects E. coli. So when they allowed this bacteriophage to infect a E. coli bacteria, so what went inside from the bacteria?
The DNA.
The DNA. So this DNA was labeled. Okay. So where the infection happened, you will find in the tube after centrifugation, when all the bacterial cells settle at the bottom, they got fluorescence signal from here. Okay. And in the supernatant, there was no fluorescent signal in case of phosphorus labeled vial. In case of sulfur labeled, they found the signal only in the supernatant and not in the cells, because the capsid of the virus, this viral capsid, which is the protein, it does not enter inside the cell. Now, if both sulfur and phosphorus were present, let me again read the question. What the question says, if both the DNA and protein have S and P, will the result still be the same? So answer is no, it will not be the same. In fact, you will find radioactive signals or radioactivity both from the supernatant and from the sedimented cells. So the experiment would not be able to prove what is the genetic material that's entering inside the cell, and you will get radio labeled signal from everywhere, because both proteins and the DNA contains both sulfur and protein. So sorry, sulfur and phosphorus. Is that clear, Abhiya? Yeah. Yes. So the whole logic of the experiment was to distinguish between the two, hence to label something which is exclusively present in one and not present in the other. Okay. That was Hershey and Chase's experiment. And just to put things in context and to give a quick revision, remember that every MacLeod and McCarthy also kind of proved the same, but their experiment was not visually appealing. It was a biochemical experiment where they degraded one by one each of the candidates and saw that in which case the Griffith's transforming principle still holds. Remember that experiment?
Yes, sir.
So in today's time, if you do that experiment with proper control and everything in place, that still would be a very, very good experiment. In that time, it still was a good experiment, but because there were more chances of contaminants and everything was not explained in proper controls, so biologists were very skeptical, and they did not, many of them did not take that result as a proof that DNA is the genetic material. And therefore Hershey and Chase has to come up with something more innovative and more logical to put to give a straightforward result. Correct? Okay. Any other doubt, people? Anything else anyone wants to ask? No questions? Okay. So let's start with today's topic. So today I'll first talk about PCR, and this is something that you all have heard about a lot in the last 3 years, especially, regardless whether you have studied science or not, or you know science or not. You must have studied RT-PCR, right? So PCRs are of many types depending on what we want to achieve, but like in the end, what we end up doing with PCR is amplify the genomic region. So it's known as polymerase, like it's PCR
Chain
stands
reaction.
for.
Sir,
Yes, Arpit.
we did the same thing in the COVID test, RT-PCR test.
Yes. So I'll tell you what is RT-PCR also quickly, because it's related to COVID,
It's
and sometimes these.
reverse polymerase chain reaction, right?
Sorry.
Uh,
It is.
um,
Yes, RT-PCR is.
what is the full form? Reverse transcription polymerase
Yes,
chain reaction?
reverse transcriptase polymerase chain reaction. So we use enzyme reverse transcriptase in that. So so there are many different types of PCRs that have been developed from this basal parental technique PCR that was initially in kind of invented and put in context by Carey Mullis, and then he went on to become a millionaire, started this multi-millionaire company because they have the patent rights for PCR technique. And there's an interesting story associated with PCR, that Carey Mullis for a very long time was trying to figure out how to amplify a specific genomic region of interest. Okay, and he knew that everyone knew that inside a normal cell, DNA is replicated. So there is a machinery inside a cell which is responsible for replicating DNA. So that can be used to also replicate and make copies of your gene of interest. But the only problem was that how to open the DNA. Now to open the DNA, if you are using the same enzymes that cell uses, then we do not understand the complex mechanism. At that time, if those molecules are always present inside the cell, so they will open the DNA. But how to close the DNA then? How to make sure after the primers get there, and let's say the DNA has been replicated, how to close it, then again open it once that replication has happened, to continue the cycle basically. One easy way to open the DNA is to denature the DNA. You give DNA heat, like heat which will denature the DNA but not destroy it. But point is that temperature is around 94 to 96 degrees Celsius, less than the boiling temperature of water, because the solution in which we do it is mostly water. So we do not want water to vaporize, and in that way, some water still does vaporize, but because it's happening in a closed tube, we don't lose much of it. But we still do not want to touch the boiling point of water. So 94 degrees, 95 degrees is enough to denature the DNA. But now the problem is You can open the DNA, but you cannot make DNA polymerase, the enzyme, which is the principal enzyme, to work, because that does not work at that high temperature. In fact, all enzymes in our body have a narrow range of temperature tolerance, which is around our body temperature, which is 36.9 degrees Celsius or 98.6 degrees Fahrenheit. So plus minus a couple of degree temperatures, enzymes work the best. That's why, have you ever felt that when we get fever, we feel very weak. We not just only feel weak, we also feel deprived of energy. We also feel very low on immunity. We also feel like our body is not functioning properly, correct? It's not just the physical weakness that we feel in fever, correct?
Yes, sir.
Yeah, that is because many of our enzymes are not working at their optimal pace and in an optimal way. But that is a trade-off that we do. That is a risk that our immune system takes, because if our enzymes will not work, the bacteria or the pathogen who has invaded our body and which has caused infection, their enzyme will also not work. So the whole point is to hold the infection, to hold the fort till a time where the bacteria, like either you kill the bacteria or the bacteria get exposed to our immune cells. We do not allow the bacteria to replicate and multiply very fast by causing fever. Okay, so in many infections, when we get fever, our immune system is active. It's causing an inflammatory response. So fever is a response of the immune system, and then we take paracetamol, turn down that response, and that's like we are working against our own immune system. So just taking paracetamol in the case of infection is not a good thing. You should know if it's an infection. You should also take something that will help you get rid of the infection. Many people just take paracetamol and they just feel that everything is fine. Everything is not fine, because you are allowing the pathogens you are helping the pathogen, not your body. Okay. So it's very important to know why you are having fever. If it is because of too much of exercise, exertion, lack of sleep, then yes, taking paracetamol works. But if it is because of a viral or a bacterial infection, then you also have to take something else to help your immune system. Okay. Yes, but we'll talk about different types. But let me just quickly take this picture from NCRT. Can you all see this picture? This is a diagrammatic representation given in your NCRT for qPCR. Sorry, for normal PCR. Can you see this, everyone?
Yes.
Don't get intimidated. It's very easy thing. So there are basically three things in every cycle. So in the cycle, this here is your gene of interest. Let's say the region of the DNA. DNA is double-stranded. 5' to 3' end is one strand, and then 5' to 3' end is the other strand. And let's say this region which is marked in red is my region of interest, RY. And this is one that I want to amplify. This is the gene that I have, let's say in biotechnological terms, this is the gene that I have isolated. In the last class, we were isolating genomic DNA, correct?
Yes, sir.
We learned a lot of interesting things about genomic DNA. Fatima must be knowing the best, right, Fatima?
Yeah.
So let's say this is the region. This is my gene. Okay. So I want this gene in millions of copies before I go on and make. What will I do next? Between isolation and after amplification, what will I do finally of this genome, of this gene? I will make recombinant DNA, right? Put it into plasmids so that I can transfer it effectively into host cells. So before that step, many a times we need to amplify it. Now, first thing is we have to denature. So we apply at this strand around 94 degrees Celsius of temperature. We heat it. So first you heat the DNA. When you heat the DNA, it opens. So you can see that the DNA has opened into two strands, the 5' to 3', and the 3' to 5' has opened up. Now we'll put some primers. So these primers are, you can already, so everything is already present inside the mixture when we put it in this cycle. But when it was first done manually by Carey Mullis, it was a tedious process. So Carey Mullis actually used to heat it, again cool down to certain things, add primers, then again repeat the same cycle over and over manually. And then he realized, okay, the DNA is getting amplified. So now everything is automated. It's machine. There comes a PCR machine which is as big as a suitcase, and you can do things in that. Even smaller than that, machines come, PCR machines come. So you heat these open and then you put primers. Now this primer, let's say if this sequence is A, G, C, A. So this primer will have T, G, C, T, this sequence. So it will go and bind. You just need to bind at the beginning, okay, and at the end. So there are two primers. One is here and second is here. So one is called the forward primer that goes into 3' to 5' direction, which is the normal working direction of DNA replication by DNA polymerase. And the other one is called reverse primer. So forward and reverse, two kind of primers are used, and it's very important to use both the primers if we want to amplify the region of the DNA. So it will go and bind. This step is called annealing. Annealing means binding of the primers to the template strand. Is that clear?
Sir,
So in one, yes, Arkat.
sir, first you can complete, then I'll ask.
Yeah. So okay. So in this strand, this will be the 5' end, correct? And this extension will happen in this direction, 5' to 3'. In this strand, extension will happen in this direction, opposite, because this is the 5' to 3'. And after this cycle is complete, you will get, instead of one double-stranded molecule, you will get two double-stranded molecules, right? So that is the whole point. In one cycle, you double the DNA. If you start with one strand, it will become two in the next. If you start with two, it will become four in the next, and so on. So annealing, still extension has not started at this point, because the most crucial thing for extension is DNA polymerase. We are just annealing it, and for annealing, the temperature can come a little down, around 70. Yes, tell me.
Our teacher told that it is 60 degrees Celsius.
You don't have to come to 60 all of a sudden. It can still work between 65 to 70.
So she
Like I
told
used
that
to,
this is like required temperature for the primers to attach.
for primers to attach, primers attach around, it depends on the primer length. Yeah, but if you want to just take a 60 degrees works, depends on what the longer the primer is. So we cannot make very long primers, obviously, because very long primers won't, first they'll be very specific, second their annealing will take time, and we also cannot take such a small primer that it can non-specifically bind to other regions of the DNA. So we have to choose between, because most of the time we do not cut exactly the gene fragment. Do you see that there are flanking fragments here and here?
Yes.
It's not advised, or we cannot sometimes cut exactly the gene out, because we have to choose the least flanking region that comes up with the gene of interest, but it does come. So we do not want the primers to non-specifically bind. So it has to be specific at the starting, and at the end of the gene only. That we can control, because that primers we are designing. So 62, 65, even sometimes 70s, depending on primer works. But okay, if it's is it in your NCRT that it's 60?
No, sir, actually, I guess that's not given in the NCRT.
Okay, so just remember that the temperature has to be lowered. Okay, it has to be lowered from 94, because at 94 the primers will not anneal, because it's the same thing. At 94, everything denatured. It opened. So you have to lower down the temperature so that things can now again come back and attach to each other. Now, one question is, if I have put primers, how are we sure that only primer will come and attach? And when we lower the temperature, it can't happen that these two strands come and attach to each other again. Does that question come to your mind? It came to my mind when I was doing qPCR for the first time, that I'm putting my primers, but it's possible, no? That
Yes, sir.
I'm raising the temperature, my strands open up. I lower the temperature, my strands close. It does not allow my primer to attach.
But then where will the primer go then?
Yeah, it will just stay in the solution. Or maybe it attaches in some molecules, but not in all the DNA. But primers attach because my gene of interest will be a very big region. Primer will be a very small thing. So when we lower down temperature, it's easier for smaller primers to attach first. That's why I said that we also have to be careful not to make a primer too long, that it's not very good. It does not show very high rate of annealing. So everything has to be optimized in research. When you study it, you study the basic. So the basic is you denature at a higher temperature, and then you allow it to anneal at slightly lower temperature. And when it anneals, next is the job of DNA polymerase. DNA polymerase done the same thing. It extends and continues extension of the nucleotide chain. But this DNA polymerase is that Carey Mullis figured out that we can use is called Taq polymerase. This Taq polymerase comes from a bacteria whose name is Thermus aquaticus. You know what's the specialty of this Thermus aquaticus, Arpit?
Yes, that this can
Yes,
withstand
please.
with high temperatures.
Why?
Because
What's special about this polymerase? Our polymerase cannot withstand high temperatures. No other polymerase can withstand high temperature. Why only Thermus aquaticus polymerase can withstand high temperature?
Is it hemophilic? I mean, sorry, heat loving?
What is heat loving? The polymerase.
Yeah.
Molecules, no, polymerase is not heat loving. Polymerase is evolved in heat because Thermus aquaticus, as the name suggests, it's a bacteria that lives in water and in hot water. The word thermos comes from temperature or heat energy. Aquaticus comes from where it's found. So this bacteria is found, it's an extremophile bacteria. Remember about extremophiles? Extremophiles are those bacteria that likes to live in environment where other life cannot sustain. For example, halophiles live in very, very salty environments. Thermophiles stay in very, very hot. So these bacteria can survive in hot geysers. Now, people were puzzled that if these bacteria can survive in hot geysers, they might also be replicating in hot geysers, right? These very heated waters. So how are they replicating there? Maybe they have some kind of polymerase or some other molecule that helps them. So they figured out that their polymerase have slightly different structure that can withstand very very hot temperatures and still can do its activity of extension or replication of the nucleotide strand. So they purified this polymerase from these bacteria, and when they used it, they realized, oh, it can be done, because still at this point the temperature is very high as compared to a normal temperature. Okay, so Thermus aquaticus can continue to do extension even at 70-80 degrees Celsius. Okay, but you need to give this enzyme some deoxynucleotides, because these deoxynucleotides are the nucleotides. Simply, the deoxy word says that it is the nucleotide of the DNA. Okay, if it is the nucleotide of the RNA, it's called ribonucleotide. Okay, it's deoxynucleotides. So this nucleotide will keep on adding one by one with the help of this polymerase. And the third step, which is extension, will happen. So you see this, that this trend started from here, but now it has gone all the way till here. So our gene of interest is covered, correct? Here also
Almost covered, right?
our gene of interest is covered. Yes, what do you say, Fatima?
I said almost covered, not completely covered.
No, it's only the red part is the gene of interest. So that's completely covered, right?
Yeah.
Yeah. So this is the flanking region, which is not our gene of interest. It just comes up when we isolate the DNA. It just comes up with that. So now in the next cycle, we have two strands, correct, of our gene of interest. From we started with one strand, now we have two strands, correct?
Yes, sir.
And if I again put all of this back into the cycle, now this two will become, through the same process, denature,
4.
annealing, extension, it will be 4. And within 30 cycles, there will be around 1 billion amplicons, 1 billion strands. Now it seems a little weird to you, but that's the power of exponential growth. So exponential growth goes very fast, up like this. Okay, so the question, that kind of question that comes in your entrance exams is, if at the 30th cycle, now listen to the question carefully, if at the 30th cycle, after the completion of 30th cycle, there is 1 billion DNA strands, double stranded DNA strands. Okay, so how many strands were there after the completion of 29th cycle? If at 30th cycle there were 1 billion DNA strands, double-stranded DNA strands, how many DNA strands were there after the completion of 29th cycle? People, just a logical question. In fact, you have to do no calculation for this question, zero calculation, almost zero. Some calculation in your head, but let me make this question easier. What if I ask you, if at the 30th cycle there were 1000 double-stranded DNA strands, how many will be at the end of 29th? Let me make 1 billion to 1000. Anyone?
500.
500. Is that correct, people? Yes, that's correct. There'll be 500. So what will be the answer for this? For 1 billion, there'll be 500 million. Okay, that's the half of 1 billion. 1000 million is 1 billion, right?
Yes.
Yeah. So if I just go one cycle before 28th cycle, there will be 250. And just one cycle before 27th cycle, there will be 125. So if I tell you that at 27th cycle there will be 125 strands, how many strands will be there at the end of 30th cycle? Just 3 cycles more, and from 125 you get to 1000. So that's called exponential increase. So you understand how at the end of 30 cycles, if we start from 1, we can reach till billion, because it just doubles. In every cycle it just doubles up. So bigger the number, in the next cycle it will be even bigger. If you
So,
start with, yes,
so I have a doubt.
yes.
Sir, this red portion is basically the gene of interest which we need, right?
Right.
So, sir, what's the need of whole process of amplification? Why can't we directly insert into the vector and make it a recombinant DNA?
We can make recombinant DNA even with few, but sometimes we want to amplify it before making recombinant DNA, because we are not, there is never 100% chance that all our recombinant DNA will be taken up by the host bacteria. Remember, we have to make the bacteria competent by heat shock, and still we have to use selectable markers to figure out which of the bacterial cells have taken up our gene of interest. So many will just be lost.
So, sir, it just acts as a reserve, like for security. We do it like.
You're just making it into millions, so that even like at least you will get, if not millions, then 10,000, 20,000, 50,000 bacterial cells which are at least transformants, so that you can grow them, right?
Okay.
Yeah. So amplification is a very, very good strategy before you make DNA, recombinant DNA. But even with few, you can make recombinant DNA, which in case, if you are actually doing an injection, let's say you are transferring that recombinant DNA inside the cell. So even if you make 50 recombinant DNA and you put it in 50 cells, the work is done, right? But
Yes.
amplifying the gene is always a good thing, because you can also keep the gene at reserve, so that you do not have to go through the whole process of isolating that gene over and over again. Once you have isolated, you can amplify it as much as you want, and you can keep it into different aliquots to use it in future. Okay.
Yes. Yes, I got it.
Yeah. Yes, Abia, you had a question. I think someone else unmuted. So is PCR clear to all of you? So it's polymerase based. That's why it's called polymerase. Chain reaction, because just like in a chain, you just continue following the same steps over and over again, and you keep on getting more and more and more and more DNA. Now, there are different types of PCRs, like one of which you have heard about in recent times, that is RT-PCR, which is reverse transcription PCR. And this was specifically done in COVID. Can you tell me why? Why specifically reverse transcription PCR?
Because it is COVID is an RNA virus.
Perfect. Because COVID is a RNA virus and COVID's genome is RNA. So if we have to amplify any region in that, we first have to convert the RNA into DNA and then do the PCR. So it's called reverse transcription PCR. There is also something called qPCR. It's called quantitative PCR, where in the real time, it's also known as real-time PCR. So people often get confused RT with real time. So qPCR is called quantitative, because you can just see the result on a screen right when this amplification is happening, in the form of a graph of fluorescence. So you will see that there is a fluorescence increase like this. So you know that your gene is amplifying right there when the experiment is happening. For others, you only get to know once you have run 30 cycles, then you run that sample, you take thoda sa sample and you run it on a agarose gel, and you see that, oh, you get a band or not. But qPCR is quantitative PCR, also known as real-time PCR, because it's happening in real time. So often people think that RT-PCR is real time. It's not. Okay. Then there is touchdown PCR, hot start PCR, all different kind of techniques, just standardized and adjusted for research purposes, whatever goal you want to achieve. But the bottom line for all PCRs is amplification of nucleic acid. Okay, is that clear, everyone?
Yes, sir.
Great. So note down things if you want to note from the screen, and let me know when you are done. And please tell me if you have any questions.
So, what is the temperature at extension? Is it?
For extension, again, extension can be around 70 degrees. Same. So when you put the primers, and the primers goes an anneal, you don't need to lower the temperature that much. You can even increase a little bit, because DNA polymerase will still work. So if you can still go around 72 degrees Celsius to 80 degrees Celsius. But if you want to, again, depending on the primers, but you want to see the standard ones, what do they take for standard? Let me check with NCERT. Okay, so it says denaturation. So one of the books says denaturation happens at 94. Then you lower it to around 65 for, I think, annealing. Okay, so this, what your teacher said, 60. It could, you can write it at 60 to 65 for annealing, and for extension, as I said, 72 degrees Celsius. So keep it 72 on this part. 94, 65, 72. Okay.
Yes, sir.
Yeah. Actually, this has a range 55 to 65, depending on, again, you have to decide it. But what is mostly fixed is this 94 and this 72. This can be a range. Take the middle value. Let's take 65 or 60, 65. Yes. Any other doubts?
So what is that deoxynucleotides written?
Yes, it is. What is a nucleotide? You know, nucleotide, it is the monomer of a DNA.
Yes, sir.
Yes. So deoxynucleotide is just the way to write that it is the monomer of a DNA. If it is just nucleotides, you can ask whether it's a monomer of a DNA or a RNA. So ribonucleotides contains uracil instead of thymine. Deoxynucleotides will always contains thymine because DNA has thymine and RNA has uracil, right? So deoxynucleotides are monomers of DNA.
Okay, sir.
So that is what the polymerase puts one after the other and extends the chain, right?
Sir.
And every nucleotide has three components: a phosphate, sugar, and a nitrogenous base, where sugar and phosphate make the backbone and bases protrude towards the inside. Make sense, people? Everything is clear?
Yes, sir.
So it's connecting your concepts of genetics and DNA structure with biotechnology here. Done, everyone? Let me know when you are done, people.
Sir, done.
Okay. So with that, let me start with some applications of biotechnology today, which is also in next chapter, but they both are kind of, if you understand one, the other is just the extension of it. But I think as chapters it was there.
Sir, I guess we left with bioreactors.
Oh yeah, okay. Yeah, we left with bioreactors. Okay, so downstreaming process you mean? Correct.
Yes,
Sorry. Okay, so downstreaming process is very simple. It just means that once you have successfully amplified the gene of interest, you have successfully put it inside the bacterial host cells, and you have also successfully um, multiplied the bacterial host cells in large numbers. Now, when we do it on a large scale, for example, to to obtain a gene product. Now, what is a gene product? Downstreaming process is done for the gene product. What is the product of a gene in biology? In general, like I told you in the beginning. Yes, I told you in the beginning that we will only deal with protein coding genes here. So there are genes that do not code for protein, they only code for RNA. And that's where in chapter, I think 6, we studied that the concept of cistron came, right? Monocistronic genes and polycistronic genes. Because we realized that all the genes do not go from DNA to RNA to protein. Some just form RNA and never form a protein. For example, what genes only form RNA and not form protein? The genes coding for tRNA and rRNA, ribosomal RNA, transfer RNA, they never go to, only mRNA goes forward to form proteins. But here the gene product is protein. Now, how do we get to, how will we get out that protein from a bacterial cell? So when we are allowing the bacterial cell to grow, we are allowing them into huge chambers that can culture large volumes up to 1,000 liters. So the culturing is done in bioreactors. They are not any ordinary vessel, but they are vessels that are technically very advanced. So a lot of engineering goes behind these big vessels, because these bioreactors have controls. So it can control almost everything inside it, every aspect of environment. So what it can control is, in one word, we call it optimum growth condition. So you can set every growth condition, which is pH. What will be the pH inside the bioreactor of the solution? You can also control the humidity if that is required. You can control the temperature. Not only this, you can also control the O2 levels, oxygen levels, and sometimes if you're working with anaerobic bacteria, the CO2 levels. Everything can be controlled. So temperature, pH. You can also control the level of salts, the concentration of salts, the concentration of vitamins and other nutrients. Everything can be controlled in these bioreactor chambers, and they are very big chambers. Even the pressure inside can be controlled. Okay, so this way we can allow the bacteria to grow under a growth condition that we have decided or designed. And while they are growing, they are also replicating our gene of interest, which is present inside them. They are also transcribing and translating that gene to make proteins. So we don't only have lots and lots of DNA of our interest inside the bacteria, we also have lots and lots of protein of that gene inside the bacteria which is growing, correct? So if in a 1,000 liter bioreactor there are, like, billions and billions of bacteria, we can get billions and billions of these protein molecules. Taking an example of insulin, this is how it is synthesized. So how will you get it out is the point. Any idea? And before you get it out, one more thing that I should tell you is that you cannot allow the bacterial cells in this bioreactor to sit, because they will settle down at the bottom, correct? So you have to constantly keep stirring or moving the solution in that bioreactor. So there are two ways by which it is done. Like, constant moving or mixing, sorry, I should use the word mixing of solution needs to be done, because if you do not do this, bacteria will keep on growing and keep settling at the bottom, and at the bottom they will start forming layers, and the lowermost layer will not get nutrition, neither oxygen, and it will die. So we have to keep all the bacteria suspended in the liquid and constantly moving around, okay, so that every bacteria gets access to everything. So that is done by constant mixing. So there are two types of bioreactors majorly in the field. One is which has a stirring bioreactor, it's called. So it is just like your mixer grinders, but it does not mixes. Well, like it mixes, but it does not harm the cell. So stirring bioreactors where they have stirrer like things in the center, and it will keep mixing the solution. The other one is where you do not want to stir the solution but still want to keep moving, is called what do we call it? It uses basically gas, so it's called sparged stirrer tank, sparged stirrer, which uses gas
The
bubbles.
first one is called the simple stirrer tanks, and the second one is
Yeah,
sponge.
yeah, simple is like just a stirring or stirrer, because if you're not, stirrer is stirrer, sparged stirrer is stirring with the use of gas bubbles. So it does not go round and round. It's just like when a solution boils. So you it's like aquarium thing. So from the base you allow oxygen to mix inside the liquid, and oxygen will keep on going, forming bubbles, going up, and in that process it will not allow any bacteria to settle at the bottom. Okay, once you have your desired outcome, desired output, or the desired weight of bacteria, or the concentration. And also remember, we cannot keep, there is a limit till which you can grow bacteria in howsoever large quantity you are growing. After one point of time, the bacterial concentration will be so high that it will not sustain with the given space and nutrition and resources. So in every bacterial population, if you have studied the graph, it's like it enters a log phase and then it gets into a lag phase. So there is growth which becomes slow, and after some time it saturates because cells also start dying now. Okay, so we have to just capture the bacterial growth in this phase where it is the maximum. And then what you do? Simple. You lyse the cell and you have to take out your protein. So the next step is protein purification. So protein purification is, write down the the protein of our interest, and this protein, because it is produced in a host which does not contain this gene naturally. So remember the DNA was called a recombinant DNA. So the protein will also known as the recombinant protein. Okay. So to get your desired protein out of it, why I'm calling it a recombinant protein? Because it contains not just our gene of interest but also contains the selectable marker, correct, the protein for the selectable marker as well. So we have to remove and purify our desired protein from this recombinant protein. And when we for NEAT, just remember, whenever we express any protein in a host, where that gene was not supposed to be present, it's called a heterologous host. So a human protein, if it is expressed in a bacterial cell, is called a heterologous host, which is expressing our recombinant protein, from which we will purify our protein of interest, the desired protein, which is the insulin. Now about insulin we will learn in the next chapter in detail, but is this part clear? People, is this part clear? Okay, this no one speaking, I'll consider it clear. Yes, it says yes. Okay, so protein purification, and then this protein, where will you keep it? You cannot just keep it in water, right, in a solution. You have to, whenever we have to make these proteins are either used for as a medicine, right, in therapeutics. So either they are medicines or therapeutical drugs, which means the same thing again. But these things cannot be just kept in water, so they have to be kept in a formulation. Formulation is a solution in which these proteins can stay stable and do not get degraded. So formulation with suitable preservatives. And this is a very important part where research is done, extensive research is done, because sometimes in case of COVID, you must have heard of side effects of vaccination, correct?
Yes.
That some people took the vaccine shot and they immediately showed very vigorous response of some or the other kind, and even some deaths due to vaccination were reported, but those were very, very few in the population. So the vaccine was given, it was not showing a generalized response in every human, so the vaccine was not generally malfunctioning. Then why few, one or two in 100, why few people? It was not also 1%, it was very less, but why these few people showed some vigorous response to um, um, uh, a vaccine? So this was used against vaccinations as an example by many, that it's better you do not get vaccinated because vaccines itself have harmful effects. It's not the vaccine that has the harmful effects, it's sometimes this formulation in which the vaccine is basically that vector containing the RNA for the gene of spike protein of COVID virus that has to go inside the cell to cause anything. But the chemical in which this vaccine is preserved, or the liquidy substance that goes inside your muscles, people sometimes with already some kind of a condition or a disease in their body, which is called comorbidities, they might show a reaction to these. Now, it was not very well studied at that time because this was the fastest vaccine that was developed to help the world come out of pandemic. But over time, these formulations become better and better and better, and hence the side effects or side reactions of vaccine go down. Okay. Is that clear, everyone? So in the beginning, they never give it to people with heart conditions. Someone, so they were always asking. So if someone have had a open heart surgery or any kind of a open body surgery or transplant or a tissue transplant or a graft or a treatment or a cancer treatment, were not given vaccines in the beginning unless and until they were 100% sure that it will not show any response or side effect. Even kids were not given in the beginning, only the adults were given. And yes, so is the formulation part clear? Then this thing goes for clinical trials. Before it can be clinical trials is where some volunteers volunteer to see any side effects of these So we use some patients who volunteer to get this treatment tested, and some normal patients who volunteer to get the side effects of this tested in a normal person. And once it passes the clinical trials, then it can be used for therapy. Okay, so clinical trials in case of medicines, in case of it's any other thing, then you need to go through quality control testing or quality control trials in laboratories. Okay, please write if you want to write anything. Now let's go to applications of biotechnology. Now, when I talk about applications, there are many applications of biotechnology. Anyone would like to tell about some applications in your day-to-day life? Starting with vaccines, correct? Anything
Yes,
else?
so it can cure a disease.
It can cure disease. Yes, it can cure
Cure
I
disease.
mean,
vegetables.
genetic disease.
Genetic diseases. Yes, you mean gene therapy. Yes, gene therapy relies on biotechnological. But this is something, vaccine is still something which you see in your day-to-day life. Gene therapy is not something that you see in your day-to-day life, but many other things present around you, you see in your day-to-day life comes from biotechnological
So there's some
advancements.
genetically modified vegetables.
Yes, so I will write GMO, genetically modified organisms in general. Now, if they are plants, they are called GMPs. If they are crops, specifically the crop plant, they are called GMCs. If it is animals, it's called GMA. Okay, so genetically modified organisms serves a very greater purpose, and we will talk about these animals later on in this chapter. But these are the organisms, plants and animals, bacteria, viruses, uh, fungi, anything whose genome has been edited or tampered by us to use them for something useful, or to give them capabilities which was not present in them before, for our benefit. Because remember, the whole definition of biotechnology was to use biology or product or components of biology to make useful products or services for humankind. So we'll keep that in mind before we do anything. Okay, apart from GMOs, something that you have already studied in this course, IVF, in vitro fertilization, correct? That's also due to biotechnology. We take gametes from human, both male and female. We make them fertilize outside the human body. So that's why the baby is given a very popular name, test tube baby. That's IVF. And not just this, something as trivial as waste management that you must have studied in microbes in human welfare. So waste management is also done through biotechnology. We culture certain bacteria which we use to reduce the BOD of the waste. Anything else? Processed food. Over the time, we are relying on processed food a lot, right? We all eat processed food in some or the other form. We should not eat much of processed food only. We should rely heavily on freshly cooked or raw food, raw vegetables, but processed food is a very important part. especially in places where it's not possible to cook food all the time. But that also happens through biotechnological advancement. So majorly, let's divide it into two groups: vaccine, gene therapy, IVF, things like that comes in medicine. Okay, so biotechnological applications in medicine is the first thing that we will cover. Second thing is majorly associated with food and food production. So GMO, processed food, they will come in biotechnological applications in agriculture. Okay, is that okay? So these are the two things that we will majorly focus on in this chapter. Now let's start with a simpler one: agriculture. I'm so sorry, people. Just give me 1 second. Okay, am I audible, everyone?
Yes, sir.
Just 1 second. Yeah. Okay. So talking about agriculture, you already know the basics of it. So tell me, why do we? So agriculture is happening from how many years now? Any idea in evolution? We talked about um, human evolution. When do we see traces of agriculture in human evolution?
Can you repeat the question?
Human evolution. When do we see traces of agriculture? When did humans start doing agriculture? Chapter 7, evolution, the last topic, human evolution. We talked about cave paintings and evidences of agriculture. Okay, if you do not remember, there are traces of agriculture which have been found as far back in time as 30,000 years ago. So humans have been doing agriculture for around 30,000 years, even more than that probably. So for 30,000 years, we were doing good. We were doing agriculture and everything was good. There was no biotechnology at that time. Biotechnology is very, very recent, in just a few decades back. Suddenly, now we started using biotechnology to do agriculture. What's the need? We were doing good, right? What is the problem that we started facing that we had to solve? And hence we thought of using biotechnology. So whenever you are studying something, don't just start remembering it. Okay, so one thing is over. Next is biotechnology in agriculture. Just step back and ask yourself, why? Why do we need to use biotechnology in agriculture? Just to look fancy? I can do agriculture, but let me do agriculture the scientific way. No, right? Everything comes out of, so they say that necessity is the mother of discoveries or inventions. So similarly, what do you think? What is the necessity of the time? What is the need of the time associated with agriculture that we could not solve just by our traditional techniques? So we had to look towards biotechnology and biology engineers and genetics. It's simple, people. Anyone?
For the wantings of the people, demands.
Why? What are the demands of the people? They all of a sudden want to eat genetically modified vegetables, and we are fed
No,
up
but
with.
like,
Yeah.
for example, like in a place, there's like
Mhm.
not much of watermelon, for example. But then
Mhm.
watermelon, this is just an example. It could be like some
Mhm.
herbs, and
Mhm.
it's not growing properly over there. So you can
Mhm.
genetically modify and get it.
Why can't just import it from where you get watermelons? We have been doing it for a very long time. From thousands of years, people trade. All of a sudden, you want to say, okay, let's stop trading. Let me grow watermelons in my Sahara Desert. You are correct. That's also one of the advantages. But I'm asking, that cannot be the need that drive biotechnology, right?
Yeah.
Coming into that, that could be one more add-on that comes with it. So don't focus on the periphery. Focus on the. I'm saying focus on the uh, fries. Periphery will come with it. You're focusing on the periphery right now. What's the bulk? Why do we need to genetically modify crops first? Why it was all going smooth and okay a few decades, a few centuries ago?
Maybe because extinction happened,
Whose extinction?
uh, of some plant.
So we are not reviving plants that are extinct. We're just growing the same plants. Rice is not extinct. It was never extinct. We are just growing rice, cotton, wheat. Have you heard of
Is
any
there a
plant?
specific uh, type of rice?
In fact, we are making, by type, let me clarify, we are making certain varieties of rice which are not naturally present at all. So we are not worried about extinctions. Rice alone has more than 50,000 varieties just in India, which are currently in the library. Library
But
means
then, so
this.
how is that a need?
That's what I'm saying. That's what. How is bringing back extinct plants is a need. That's not a need. Again, you are focusing on the periphery. Focus on the
No,
fries.
I think it is a need. I mean, because, like, for example, if just an example, if rice plant is getting extinct and you need
Rice plant
rice.
is not getting extinct at all
No,
anywhere.
I said, for example, like if there's like a very medicinal plant which is going to get extinct, you can
Mhm.
genetically modify the genes and get the plant.
You No, you cannot genetically modify a plant if it is extinct. You need the plant to genetically modify it. If it is if it is on the verge of becoming extinct, you don't need to genetically modify it. You can just grow it in protected environment like we do for animals which are vulnerable to extinction. So for example, kangaroos, if they are vulnerable to extinction, or tigers, which are, if they are vulnerable to extinction, we don't change their genome. We just protect them. We do not allow their hunting. We do not. For plants also, we just grow them in large numbers, and then put them back in forests. Go check on them on a regular manner to just increase their number. If they are extincting, we do not need to change their genomes. But here we are talking about agriculture, biotechnology in agriculture. So basically the plants that we are talking about are agricultural plants, which means they are crops. Crops are not getting extinct. So why are we doing this? Because it's 21st century, we need to do something in a cool way. Okay, so I think you have reached maximum of your. So sometimes what happens is we think so deep that we miss what is very obvious in front of us. What is obvious in front of us is that few centuries ago, the human population on the planet was very less. In just last 200 years, we have we have increased around 16 folds, and we are now 800 crore people on the planet. And the land for agriculture is limited. We cannot get as much land as we want to make it agricultural fields, because we have been doing it for a very long time and we have been tampering with the balance of ecosystems. We have been clearing forests, making cities. We have been clearing forests, making agricultural fields. But now we have reached a threshold where if we continue doing so, we will, tamper with the balance. And if the balance of ecosystem goes, then we will see mass extinctions. We will see climate change ramping up and things like that. We will see animal human interaction. We will see diseases like COVID coming up really, really fast. And because all of this happens when humans invade the animal's territory and start living in very close connections, close proximities. So many of the viruses come from bat and pangolins for a reason. Bats and pangolins are good reservoirs of viruses, but they do not live around human very much. They live in caves, in dark, damp, isolated places. But then we start going invading their territories, growing our plantations of fruit and things. Then these bat comes, eat those fruits and in the night, but does not eat it completely. They just like eat it, leave some viruses in that fruit. Those fruits are eaten by other organisms, including humans. And these continuous interaction between animal virus and human virus allows the virus to switch host, jump from one organism to another, which was not a host for that virus in the beginning. So this COVID-19 is not a new virus that emerged in 2019. This virus is present from, like, I don't know how many millions of years already. It just started attacking humans and started recognizing human cells as host. That's all. So the problem is we have limited land. Coming back, so the root of every problem, almost every problem with ecology, is growing population of humans. So we have more mouths to feed than we have food, or we have land to grow food on. So what do we do? Sorry. And then we cannot we can no more continue doing agriculture the traditional way. Sorry. So we look towards science and we start developing plant or crop varieties which are called HYV. HYV is high yielding varieties of crops. High yielding variety of crops means you agriculture the same land, the same area of land, but in that same area of land, now you will get more yield. Is that clear, people? Like per hectare of land, your yield is doubled or tripled. That was done in India during the time of Green Revolution. So you know about Green Revolution, people? It's called Green Revolution because our crops, we succeeded, the scientists along with the government and the farmers succeeded to triple the food supply and the food yield, okay, of the country from the same amount of land. But still, even if you triple the food supply, you know that in few decades the population is going to get doubled or increase similarly. So you are never producing enough food, so you have to continuously keep thinking of better and better and better ways, and you continuously keep forming new and better varieties. Now coming to the peri, so the bulk is that we have to increase the yield of agriculture. That's the main motive. Is that clear, Fatima?